All right, hello everybody and welcome to week 4 of genetic engineering as part of the case studies and science literacy series. So today we'll kind of wrap up by really addressing your thoughts and concerns. And really what I wanted to do in the first 3 weeks was talk about how the technology worked, what the current set of technologies are as we're applying them, and then what are the things that we couldn't do. And really check out episodes 1 through 3 in this series if you're interested in those topics. Today I wanted to move into what we hear. What are the things that people have told us? What are the things we've heard from others that give us concerns about these technologies? And then what I'd like to do is at the end give you some ideas about, so first I'd like to kind of do that debunking. I'd like to tell you what is myth and what is fact. I'd really be excited to hear your comments and feedback and address your concerns. And then at the end I'd like to talk about what are the best ways for us to talk to friends and family about this, because there's some tricks that we've learned over the years that really do help us become better teachers in this controversial area. So one of the things that is the big problem is, who do we trust? And when you look online, it's impossible to understand. As somebody who maybe doesn't have a scientific background, or someone who's just concerned about what they're feeding their family or feeding themselves, who do we listen to? Who do we find as reasonable and acceptable authorities? And this is really the big problem. The internet today makes it very difficult for us to know what's real and what's not. That very reputable sources, what appear to be reputable sources, say some sort of international organization of responsible technology or something, really is a broom closet in Iowa that where somebody sells books and documentaries. And so it's really hard to tell what's real and what's not. The other big problem we have today is this phenomenon called predatory publishing. And what this is, these are basically journals where you can submit some kind of soft science and along with a check and actually get it published. And this is another real big problem because it really kind of clouds the atmosphere for non-experts to be able to separate what's real, what's not. And in this important topic, it's really a problem and it's increasing. So what I wanted to give you some ideas about today were what are some legitimate questions and how do we answer them, especially because some of us have friends and family that have these concerns. But the first most important thing is, how do we really come up with some guidelines of what we should be trusting? And I think when we go back to it, there's 2 pillars that science really sits upon. And that is this idea of peer-reviewed literature, that a claim is vetted by multiple people, usually people who are in your discipline who may not share your viewpoints. And sometimes it's people who do share your viewpoints. But and those are the biggest critics. The people who like you and appreciate your work are the ones who don't want you to make a mistake. And so during the peer review process, we submit our work to others to review, other academics, and they go through it very critically and make the points they want to make. So that's what something has to go through to get published, except in these predatory journals. They may just have an editor who looks at the abstract and cashes the check and publishes it. Some atrocious work is present in the predatory literature. But the other side of this is reproducibility. Even if something is peer-reviewed, it should become part of a growing tapestry of science. That a one-off study, such as most of the anti-GM ones are, they tend to be single studies that never are reproduced or never expanded. That someone will make a claim that never grows. Science is always a race to be number 2. So if I read something really exciting in the literature, what I do is think about ways that my lab can lend its expertise to solve that same question or answer a new tangent associated to that new area. Good science expands fast. And so when we see something come out in a kind of a shoddy journal that nobody really cares about and nobody really follows up on, we tend to give that work not much credence. Additionally, even sometimes people will publish a report and that laboratory won't even follow up. That's pretty strong evidence that there's nothing there. But what I'd like to do today is really go through, start with these basic ideas. What do we trust? What do we know is real? What do we know is not real? And I'd like to go through some of the common questions and misconceptions and give you some ideas about how you can be a better critical thinker about these technologies. So the first one we'll talk about, here's an easy one. All right, let's see, here we go. Genetic engineering is the most important strategy in crop improvement. And, think to yourself, is that true or is that false? Most important. And the first one really is false. traditional plant breeding really is what's giving us the majority of traits in a genetic background. That it's not about the genetic engineering trait, that's just one extra trait in a very complicated background. That really the most important way that we can genetically improve crops is through this idea of traditional breeding and selection. That's the important background where we add in one extra trait. So that's a really important first step, is to just kind of let the air out of what this technology is and isn't. The next question I want, it comes from a really busy couple of weeks. GE crops do not yield better than non-GE equivalents. And just kind of think to yourself, is that true or is that false? Are they the same? And why this is really surprising is because there was an article that came out 2 weeks ago on October 29th in the New York Times. And we'll go through that and show you some of the claims. But the answer to this is you can't make a sweeping statement like that. Every crop is different. Every trait is different. Every place you grow it, every pressure it's under is different. And so for us to make a sweeping statement about yield would really be rather disingenuous. Yet someone did that. In the New York Times, very authoritative resource, says no, that there is no increase in yields, that this is, it causes, as they say, has not accelerated increases in yields or led to an overall reduction in the use of chemical pesticides. The technology has fallen short of its promise. And this makes me wonder, okay, we've been using this technology, or I should say farmers have been using this technology for 20 years in crops. The seeds cost a whole lot more money. It was one of the most rapidly adopted technologies. That the graph here on the right shows the adoption versus time of herbicide-tolerant soybeans, cotton, and BT, which is the insecticidal cotton and corn, shows their prevalence since 1996 when they were released. And farmers were faster to pick up these kinds of enhanced seeds than cell phones. This was the most rapidly adopted farming technology in a long time. So it costs more, and it's rapidly adopted, and an author in the New York Times says that this does not improve yields, or that it doesn't work, it's failed technology. And the first thing I thought is somebody better tell the farmers. It turns out a couple of things. Well, first is that farmers are probably the most shrewd business people out there. And if they're not making money, they're certainly not going to buy the seeds the next year. It's been true with any hybrid going back to the 1920s. So just right off the top, we can kind of tell that's probably a soft claim. But on top of that, there were no traits ever installed to increase yields. Nobody ever installed a trait to increase yields. Here you see herbicide resistance and insect resistance. So these weren't made to increase yields, they were made to save the farmer money and allow farmers to use other strategies to be able to control some of the pressures on crops. And from that, maybe you would see some increase in yields. But yields were never the target or never the improvement. We could improve yields. The problem is nowadays, crop prices like corn, soy, cotton are so low that if we improved yields, we'd make a price structure problem even worse. But here's really what it boils down to. The technology was not made to improve yields, it was made to ensure yields. And this graph shows that very nicely. That here are two lines. The dotted line is the non-GE crop. The GE crop is a solid line. And what you see in the top panel is, under these circumstances, the grain yield from this crop was about maybe a third less than the non-GE crop. That year there might have been pest pressure or some other kind of issue that made that difference very strong. 2009 they were the same. And that's very common. All you're seeing is that in 2009 maybe the pest pressure wasn't so strong. So you didn't see the advantage of the insect resistance trait. So when you say that there's a failure to yield, or when you read that, you have to keep in mind that there's lots of nuance. It depends on the crop and on the year. Of course, when you go back in the data and examine large sets of data, you can find examples that really do show very clearly that you either have the same yield or an increase in yield. So when this work on the side here on the left is from Fernandez Corneo 2014. and gives us extensive table that is a review of the literature where they tested herbicide-resistant corn, cotton, insect-resistant. And what you see is that for the most part, it's either the same or increases. And if you dig around, you can find some cases where it was actually lower. But the point of the matter isn't about yield. It's about how it protects the crop so that the farmer has fewer inputs. And when you go over and look over at this table on the, or the pie charts on the left, on the right rather, it examines farmers' reasons for adopting genetically engineered crops and looks at soybeans and corn and cotton, the herbicide-resistant and the BT, or the insecticidal. And this light blue piece of the pie, which is kind of bigger piece of the pie, is increased yields. So even though the data tell us that yields are increased and farmers buy it because of increased yields, even though there was no gene to increase the yields, it tells us that this is probably doing what we thought it would do. And surprisingly, the New York Times author got it wrong. It's important because reporters and good sources that we think are good sources, like the New York Times, can make some pretty severe errors. And it's important for us to keep that in mind. And I won't read through these, but certainly there's been good peer-reviewed literature that says that there is a clear increase in yield or flat, and that even the National Academy's report, which is a very important a report that came out in 2016, said many adopters of genetically engineered crops have experienced either lower costs of production or sometimes higher yields or both. And so it really is about how do we do something for the farmer that makes sense. Lower the costs and get the same yield, that's pretty good. Lower the costs and get higher yields, that's good too. Have higher yields at the same cost, that's good too. But the basic idea is it's something that benefits the farmer, and that's why they use them. So when we read the article in the New York Times that says that they failed to yield, we have to keep in mind it's not a simple question. Yield is a complex trait, and all of the data suggest that it actually does do something good. But the same article says that the crops use more pesticides. And if we look in that article, it says that the use is skyrocketing of an herbicide. And he even says uses more pesticide. But what is a pesticide? Well, a pesticide kills pests, right? And the problem is when we look at the literature that's critical of genetic engineering, we tend to see herbicides, pesticides, and fungicides combined into one bucket of pesticides. And so what it's important for us to keep in mind as critical readers is what are the relative amounts of herbicides or insecticides or fungicides used. We can't just lump it all together because it gives us a skewed statistic. So do they increase insecticides? And this is where you break out insecticides from pesticides in general. And these data come from, in the blue arrow I added, it shows when the GM trait was introduced for BT, the resistance to insects. And what you see is that overwhelmingly over time, the trend is significantly lower amounts of kilograms of insecticide used on cotton and on corn. This is 1995 to 2010. And again, that's widely accepted that the BT trait, the plant that makes its own protection from insects, decreases the amount of insecticide used. But this is where it gets funny. The author says pesticide increased. I showed you insecticide decreased a lot. So how do they get pesticide increases? Well, this is how they do it. They look at the glyphosate pounds applied. So glyphosate, as you recall, is the herbicide that's associated with Roundup Ready crops or glyphosate-tolerant crops. And what you're looking at here is the blue arrow is when this is introduced. And what you see is that on the bottom, the lines, the green, blue, and yellow line, talk about the amount of herbicide use in terms of total pounds on corn, soybeans, and other GM crops. There absolutely is an increase. But at the same time, there were very few acres in 1996 because it wasn't invented or released yet. And so you might expect that as farmers grow more acres of these crops, you would use more herbicide on those crops. I always make the analogy that we didn't sell much flat screen TV cleaner in 1996, but its use has skyrocketed since. Well, of course. There was none then, and now there's a lot of flat screen TVs. we didn't use much of it when there was nothing to use it on. And the author here kind of mixes that together to create some confusion and maybe some misinformation. The red line talks about total agriculture use, and that's important because glyphosate is not just used on genetically engineered crops. It's also used on grains to dry them down, meaning to make uniform drying at the end of the season. But it's also used in places like row middles and groves and orchards. They use it to kill weeds everywhere. It's used in municipalities along fence lines and in municipal use. So this amount of glyphosate applied is absolutely going up. Absolutely. It's cheap, it works, it has low toxicity. And so this is really kind of a deceiving graph to show somebody and say, well, look, obviously these crops are causing this huge increase in the amount of pesticide. It's causing an increase in the amount of herbicide because it works and there's more acres to use it on. This graph is important because it shows the relative amount used per acre on soybeans, corn, and cotton. And really what it shows you is that, sure, there may be a little bit of an increase from year to year and maybe even a trend over time. There is the resistant weeds we talked about maybe in week 2. And that reminds us that sometimes they have to use a little bit more per acre. But the general idea here is that the amount used is generally kind of flat. It's not a gigantic increase like people like to say. Yes?
So if you compare like the last slide, would you see also like an increase, like I guess from like 92 of the like herbicide resistant weeds? Would you see like also like it increasing, like correlation of it, like increasing the number of weeds that are herbicide resistant?
Yeah, so the question was, if we went back before glyphosate and talked about resistant weeds, would you see an increase in the number of resistant weeds? And you do see an increase in the number of resistant weeds. They've been resistant to every mechanism that we've thrown at them for herbicides over time. Every herbicide we've ever presented has caused evolution of resistant weeds. Glyphosate's no different. If anything, it might have taken a little longer for glyphosate to really develop the same number of resistant weeds because of its mechanism. But there are 9 ways to be resistant to glyphosate, at least, and probably more to be discovered. It's we're very strong selection, and we're selecting for tolerant weeds. So the idea of this one was basically that, yeah, we are using maybe a hair more per acre, but it's not this gigantic use in pesticide as the author claims. That we had an increase in herbicide per acre. We have more acres of the plants being grown. So, of course, more pounds of this, more kilograms being applied. Another important point of that paper, though, is when he compared the U.S. to France. And he showed these 2 graphs. And here you show France on the left, where it talks about insecticides on the bottom, herbicides and fungicides, and says, look, the trend is going down. And on the other hand, the U.S. on the right, oh, look at that. The trend is going up with herbicides and insecticides, fungicides kind of flat. What the author neglects to tell you is that, and this was taken apart by a professor named Andrew Kniss at University of Wyoming, is that if you put the French numbers and the U.S. numbers on the same graph and normalize it to the number of kilograms used, you find out that France is using a whole lot more than we are. So the author put this down like, look how wonderful France is and how their policies and general attitude towards this technology are doing good things for pesticide use. Not really. When you combine them all together, which I don't think is a fair treatment to do, actually we look pretty good. We're using a lot less fungicide and insecticide than France is. Of course, we always want to use less, and that's really important because no farmer goes into the field and says, how much can I spray today? I mean, it's a question of what can I get by with, and so it always should be something we strive to use less. So the bottom line is, again, that when someone tells us that the crops cause more pesticides on the front page of the New York Times, we have to look at the nuances. That these crops have led to us using fewer insecticides, more pounds of the herbicide because there's more acres of the herbicide, and that the other important point is that the old herbicides have been switched with the newer herbicides, which have lower toxicity, lower environmental impact. We talked before about this one, so I'll go quickly through it, but true or false? GE crops make pesticides. And we see the scary imagery all over online. And it's true, they do make pesticides. That's the idea. That's why they work. That they make a protein that, that is toxic to some insects, some lepidopterans, so moths and some butterflies. And, and that's the beauty of it, is it's specific. It allows for that decrease in insecticide use that I showed you in the previous slides. What I'm going to do is zip past this next one here and just remind you that there's a place in which this works. It's a protein that's in the gut of, that is a receptor, the cry protein, or the BT protein, is a small protein that's produced by bacteria that was moved into plants that, when ingested, has an effect on the gut of the larval insect that's chewing on the plant. And this is why it works so well and why it's part of a very important pest management strategy for organic growers. They use this stuff all the time. Only instead of having to apply it, that scientists were able to install this into the plant, so when the insect comes along and chews on the plant, whether it's cotton or corn or the brinjal, the eggplant in Bangladesh, that the insect gets sick and dies. And so it's a way of helping the plant protect itself from insect predators. So the idea of this being a myth that, plants make an insecticide, it's not a myth at all. They do. And GE crops make an insecticide that works very well against its targets. Here's one that came up a little earlier, Terminator seeds. I bet some of you have heard of this one. Terminator seeds are used all the time and destroy all of farming. True or false? Well, that one's kind of false. The idea was back in the mid-1990s that you could use strategies to control gene flow. That if you're going to be making genetically engineered plants, you want to have a way to control where those genes move in pollen and in subsequent generations. You want to be able to do it for two reasons. Companies want to restrict their technology from moving everywhere, and they also want to protect the environment from genes that are moving in places where they shouldn't be. They constructed some ideas, or at least thought about it. It actually was a scientist at the USDA who drew the diagrams for this in a book. It never was done, never was even tried as far as I know. And it was done by a company called Delta Pine and Land. And when they were purchased out by a larger company, they were later decided that they would never develop this technology. So the Terminator myth is debunked. Here's a good one. I like this one. True or false? Glyphosate is in rain and air in dangerous amounts. And this is what we see online. That it's in 75% of samples. That it's showing, here they show the child's hand, holding the mom, and, the ominous molecule in the clouds. And I remember the night that I saw this online, I thought, this is crazy. This is a new paper. And when I read the paper, the abstract online, it said, yeah, glyphosate is in 75% of samples from the air, and that were sampled. And I thought, this is great. I got to read the whole paper. I wrote to the authors. They sent me a preprint that nobody else had asked for. So the news sources of this never read the paper. I read the paper, and it said, yes, they were found in 75% of samples, but found in absolutely tiny, vanishing, barely detectable amounts, a few meters from the side of a cotton field where it was used. So these ideas that it's present in all of our environment are really a stretch. And when you look at the data from that paper, they compared 1995, where it wasn't used, to 2007, when it was used. And what you're looking at is across the season, from April to September. And the bars represent different herbicides that are used inside that field. And what you find is it's a shift. You shift from other ones, which they don't even list here, is mostly other herbicides, to glyphosate and some others that are much less toxic and have less impact on the environment. So the idea was, yes, they did find it. And if you look at the numbers, nanograms per cubic meter. So nanogram is a billionth of a gram. So this tiny amount of stuff inside the area of a cubic meter, it's really a testament to our ability to detect almost nothing. So this looked like a very favorable trend. So it is present in the rain and air next to a field, but it can be detected in extremely small amounts that are not relevant to physiology. Another common one that we see is GE crops have formaldehyde and glyphosate and have no nutritional value. This one comes up all the time. And where it started from, I got an email from a friend who is very skeptical of genetic engineering and says, aha, this corn is poison. There's no nutrition. Sends me the email. I said, okay, I'll check it out. And he took me to this table from Moms Across America. And you don't have to look at the numbers real carefully. You can Google this and you can find it. It's look up stunning corn comparison. And what they say is here's genetically engineered corn next to non-genetically engineered corn. And long story short, it's a table from a soil test. So it's not for use of biological material, the things they measure. But then what's worse is that the numbers are completely made up. They make the mistake of not first consulting someone who knows what biology is about before they populated this table with numbers. Where really, some of them are a real tip-off. I could go through it, but the basic idea that there's that it says that non-GMO corn has let me see, where's carbon? That's a good one. Long story short is that there's 60 some parts per million carbon when plants are completely made out of carbon, and 2 parts per million carbon which that's not biological. So they made the mistake of doing this, and of course when I asked them to clarify, they banned me from commenting on the site. And I wrote an email to them saying, would you mind reproducing this and I'll pay for it, and we'll do it. And they were first very excited about it until it started to get real. We started to talk about what seeds we would need, what seeds they use to do this test, where we could plant multiple tests through multiple places and get everybody on board. And then all of a sudden they backed out. So but this was something that people hold in high regard. It fails both of our tests, that it's not peer reviewed and it wasn't found it hasn't been repeated. So it's a misleading table with manufactured figures, and that's really a problem. Another one that we see all the time now, and this is coming up more and more, glyphosate is found in breast milk, wine, and beer. And nowadays, if you look online, you'll find that there are groups to be claiming to find it everywhere. And when you look carefully at this, what you find is they're using a test that was designed to be used with water and claiming that they're finding the chemical. And this is a good tip-off. It says glyphosate found in organic wine. Organic growers do not use glyphosate. They can't. And so if you're detecting it there, it kind of says that your negative control is coming up positive. That's what it says to me as a scientist. To them, look at how widespread the contamination is. So just as a way that you're going to misinterpret what is really a bad test. But here's how it works, and I won't go into the gory details, but it's what is called a competitive ELISA. And it's done by it's a kit that's made by a company called Abraxas. It's made for use with water. They say it's not a great way to use for quantitation, but it's a good first test. And then you follow up with more sophisticated tests like LCMS and other better ways to detect this compound that are much more difficult. And I don't want to go into the basis of the assay, but the bottom line is it relies on a chemical reaction to occur that could be clouded by chemicals in the sample you're testing. Breast milk is a very complicated substrate, or matrix. Wine, very complicated matrix. Any chemical in there that interferes with any of the parts of this assay will cause a false positive. And that's exactly what they're seeing. So glyphosate is found in, no, glyphosate is not found. It's really detection. What they're calling detection was noise from detecting something, allegedly, from an incorrect use of a kit. And we see this all the time. It's an inappropriate assay done by non-scientists, again, published on a website. And it tells them what they want to see. This, again, is Moms Across America. So it tells them what they would like to see. And so they're happy to publish it and scare people with this information. This is something I made here that they're talking about 1 part per billion that they're afraid of in the wine, of something that some people call a carcinogen that doesn't really have evidence of that. Yet ethanol is a carcinogen and is present at 130 million parts per billion, and then they get nervous about that. So here we go. True or false? This is an important one because we hear all the time about how 270,000 Indian farmers have committed suicide because of having to buy the seeds. And when you look at the statistics for suicides in India, you do find that it is a problem. There are a lot of people in farming environments and in cities more frequently now who do commit suicide, especially associated with indebtedness. And if you look at the trend over here on the right, what you're looking at is the cotton yields in India. So from 1980, you see the bump, how it increases very quickly once there's the introduction of the genetically engineered cotton, the insect-resistant cotton. And if you look at suicides on the bottom, suicides are shown as the black squares on that line, and the gray squares on that line, and then cotton production on the bottom with the triangles that increases very strongly. And what you see is that generally the trends are pretty flat. It doesn't seem to hold. And if you actually look at the economic data, where lots of folks have looked at this carefully, you find that actually there is a very positive effect of these crops towards gains in income in these homes and gains in yield, and generally a net benefit. So again, it's people who continue to perpetuate these discussions that don't have a lot of scientific merit. They don't match the data, and actually the data are actually quite contrary. This is a real common thing that happens. 400% increase in allergies since genetically engineered crops were introduced. And oh, look at autism. It must be caused by glyphosate. What they're showing here is glyphosate use in the red line, and yellow bars, autism detection. And you see this one everywhere. In both these cases, I'm not sure that 400% increase in allergies is wrong. To say that that's due to genetic engineering? So there's no evidence that connect the two, except for the fact they happen at the same time. But that in no way says that genetic engineering causes allergies any more than the allergies cause genetic engineering. The reason for allergies is being hotly discussed and is of great scientific interest. But the major scientific community that's analyzing the allergy phenomenon is not connecting it to genetic engineering. You think about every trait is different, every crop is different. So it'd be very strange if there was something about that. The one on the right here is really a problem because first of all, the number of autism cases is very disputable. More people are being diagnosed earlier and properly and being placed onto a spectrum that recognizes this disorder in many different forms. So numbers increasing may not be indicative of increased prevalence as much as our improved surveillance. And again, connecting that with glyphosate, yes, glyphosate use is increasing because there's more acres of glyphosate, but the amount that's present on food is almost nothing. It's parts per million in the high end, which is physiologically irrelevant. So you don't get enough intake for it to really matter. So this is really just confusing correlation and causation, and where there really is not an association other than they happen at the same time. What about GE crops cause tumors? And a few years ago, we saw a lot of newspaper headlines about a study that came out in a peer-reviewed journal testing or making a claim that these animals fed these crops developed severe tumors. And you might remember this next image. And this was the major take-home that really affects a lot of people when they look at it. Remember these pictures of these mice, that it says fed GMO, not fed a trait in a crop. It uses the word GMO, and that should be a tip-off. And then plus the herbicide Roundup plus Roundup. And what they're saying is that the herbicide or the food cause exactly the same kind of manifestation of tumors. What's missing from that? So fed GMO feed and herbicide, and fed herbicide. What's missing? Control. If you go look at table 2, you find out that that rat got tumors too. So when you're an author team and decide that we're just going to kind of conveniently leave that one out of the picture, it should be very, very clear that there's some other agenda here, that that something is wrong. And there's many criticisms of this paper. It actually was retracted and then republished in its original form in a lower tier journal without further review. And there are a lot of criticisms of this between very small sample sizes, detection, a lot of questions. Some of the data even suggested that glyphosate made rats live longer. So it's very, very shaky, but still scared people to death. And if you talk about the change in public perception just because of these rats that show up everywhere, it's important for us to keep in mind that we need to keep a critical eye on this topic because of this kind of misrepresentation of science. This is a good one. You may have heard of this, that there's a deadly organism that's present in genetically engineered crops that causes cattle abortion and diseases in humans and plants. That it causes asthma, allergies, autism, ADHD, Parkinson's disease, Alzheimer's disease, Morgellons disease, and a whole bunch of other things. This was a claim by a scientist. He sent a letter to the U.S. Ag Secretary, Tom Vilsack. Dr. Don Huber he's an emeritus professor from from Purdue, and people say, his words are taken very seriously because of his credibility as a scientist. And he stepped out into public fora all the time. He travels quite a bit talking about this organism that can be found all over Roundup Ready crops. It can be cultured, that it obeys Koch's postulates. And he calls it a microfungus. It's allegedly a new kingdom of organism that has not yet been defined by science. And he made these claims at a talk I was at. I saw him live in town here. And he showed pictures of aborted cattle and showed pictures of sick people. And the audience was just like gasping because they were so disturbed by the fact that this could happen. And at the end of the session, I asked him, I said, I can help with this, that my laboratory works with sequencing and we can sequence large-scale genomes and assemble them. Give me a sample, if you don't mind, of your organism, and we will sequence it, and we will end this crisis. And he spent the next 20 minutes describing how it doesn't have DNA or RNA, that it's being studied in labs all around the world, and that he'll publish it shortly. That was 3 years ago this week, or 4 years ago this week. It was a while ago. So, but this is another really sad story because here's somebody that has great credibility who's out scaring people with science. And and this is why we have to take these things seriously and talk about them. The other question is about regulation, and that people do say that these crops are not tested or regulated. And we see this all the time, but actually they go through a very rigorous testing and regulatory system. It starts with the FDA, where they're tested if they're safe to eat. Then you go to EPA to make sure they're okay with interacting with the environment. And then USDA to determine if they work well on a farm without invasiveness and that they don't have other problems with contaminating nearby crops or nearby wild species. The process says here on average 13 years and $136 million. So it's a very rigorous process that the FDA, and EPA, and USDA simply oversee the process. They don't do the testing at the FDA. They tell the person wanting to release the crop what they need to test. And it's not always straightforward. It's after you get the whole report back and do all the experiments, they want one more set of experiments, one more set of experiments. It's gotten much better in the last couple of years. But what you'll hear people frequently say is, there is no mandatory testing, or the FDA does not test. And they're right. It's not mandatory. There's no mandatory schedule of things that you have to do. It's on a case-by-case basis. Crops are tested based upon their apparent impacts. And a good example is here, that this apple that is produced, this non-browning apple that'll be out this year, it never had to go through EPA because that apple is grown and it's a natural trait. It's a trait that we already have in non-browning grapes. So not something that should be tested or requires regulatory oversight in that regard. But it does still get tested by FDA and has been. So that one's coming out. So it's hard to know what to believe. You have to think about everything critically, analyze peer-reviewed evidence, that good science always continues to grow, and that one-off papers happen all the time in the world of the folks who are making claims against biotech, and that we have to be very careful about what we see on websites and really count on the peer-reviewed literature. So basically we're at the end of this section of this course, and it's hard to know what to trust. Even what looks very reputable can have some major flaws. and that peer review doesn't always get it right, and that not every journal is created equal, that good science will continue to grow and continue to expand. And one-off studies that kind of die in 1997, 2007, 2012 with no follow-up really look at very critically. And that the internet, well, we all know it contains a lot of bad information. We got that. But the bottom line is that all of these sources of poor information have an effect on people. They, as I wrote here, they fuel confirmation bias. They give people the information they want to see. It gives them information that confirms their beliefs. It confirms what they already feel is true, even though it's not. And all it does is it spreads misinformation. And that's why we always have to be careful to check our own biases, right? When we're looking at these things, pro or against any of these topics that are presented inside this course, it's important for us to check ourselves for self-deception. Are we, here's what I believe, we got to check it rigorously to make sure that we're right or that we're not wrong, more appropriately scientifically. So I'd like to spend the last 5 minutes on an important part of this discussion. We talked a lot during my section on agricultural technologies, things that looked very promising. But there's a lot of scare and a lot of skepticism, and how do we fix that? If there's one thing I hope I showed you maybe last week in episode 3 was there's a lot of very good things we could do with this technology that's not getting to the people who need it. And how do we fix it? How do we bridge innovation to application? How do we make this go faster? And the big trick is we need to be talking about it. And as people who are interested in science, people who are just taking this class, maybe watching at home, it's important for us to realize that the more we inform ourselves on these topics and the more we share that information with others, the sooner those good technologies get to the people that need them. The problem is that scientists and ag producers, the people who are involved in this stuff, they just don't do this. Scientists don't step into this discussion, very rarely. And ag producers the same. They don't want to have to go out and defend the technology that they find helpful. The problem on top of that is when we engage, we do it wrong. People are just interested in taking care of their bodies and their health and their families. They just want to make sure they're healthy. And there are people out there who will use a message of fear. And whether it's Dr. Oz or the Food Babe or these folks, they'll come out with fearful messages that aren't necessarily based on science, that have a profound effect on people who are just worried about what they're eating. On the other hand, scientists and farmers, we screw it up. We either ignore them, or when we engage them, we do it incorrectly. We give them facts and figures. We give them statistics and graphs. And if you're talking about the moms who are worrying about their kids and they're getting a message from the heart of beware, this is poison, or a scientist saying, well, let me show you figure 3, you can see which one's going to win. You know, that as scientists and as people, enthusiasts, people who really want to spread a solid message of science, we have to be talking to people in a different way. And we have to think about better ways to discuss these topics in a way that resonates. The main idea is to talk to the people who are in the middle of this discussion. Don't spend your time trying to convince someone who can never be convinced. Someone who's unteachable is a waste of time. Talk to people who just want to know the facts, who want to know the evidence, who have a common interest in things that you're interested in as well, worrying about their health, worrying about their families. Make sure you understand their concerns and look at their evidence carefully. They may show you the 3 lumpy rats. You have good evidence now on how to refute that argument. Ask them for the control. Ask them to look at table 2. But actively listen to why their concerns are their concerns. And then make sure that you lead with your ethics. Make sure that you're talking to them first about what concerns you. What's your authority? What's your training? Why are you a credible source of information? So the idea of defining that audience, listening to their concerns, and then talking about the things that concern you and the reasons that you're a credible source of information, that's how we help to move this. Your audience doesn't care about facts. They don't care about the evidence. A lot of people who are concerned aren't making up decisions based upon data. You can win them back by starting with your shared values. So as communicators, it's most important for us to start with the things that are important to us. And that cuts across all areas of this course, whether it's evolution, climate or vaccines, or genetic engineering. What are your values? What's important to you? To me, it's about the environment. It's about farmers and profitable farmers for the 1% that feed the other 99%. It's about consumers having healthy food and something for the needy. And if we start with that and say, here's what's important to me, here are some ways that genetic engineering can help to solve those problems, that's the way that we can move that innovation to application. So this is the end of this section. I hope that I've convinced you that there is an amazing safety record with some very real environmental concerns that we can solve. It's just a few crops and a few traits, and that a lot of great ideas are not being used because of a fearful message that's being propagated by a very small number of people. There's a tremendous amount of misinformation, and it's really hard to understand what we should trust in terms of real peer-reviewed work. But we can do it if we think about it critically. I hope you do listen to the podcast. I think there's a lot of really, really nice episodes on there that talk not just about genetic engineering and the good things it can do, but also about the concerns and the ways that we can mitigate those risks. It also talks about crop domestication and the way that we've generated the plants that we enjoy the most. And as a final homework assignment, learn who Norman Borlaug is and study what he did. The man who fed a billion people, that's billion with a B, like Borlaug, fed a billion people. He's a good person to know, and it's really inspirational to understand how he handled the questions of world hunger and made tremendous contributions. So were there any questions today? Any questions? We have about 5 minutes left. Any other thoughts, questions? Borlaug, B-O-R-L-A-U-G. Nobel Prize winner from 1970. Just passed away in 2009. But a very strong proponent of biotechnology as another way to feed people. He was the father of the Green Revolution. Was able to identify very wide crosses that allowed him to bring in some unique genetics into Mexico, into India, Pakistan, places that were undergoing tremendous poverty and food shortages. And by being able to bring in novel genetics, was able to increase production substantially. Yeah, good guy to know. And if there's ever any questions, you know where to find me then. I guess we can end there. Thank you very much for coming.